camp there just a minute, because if evolution's going to work, there has to be new information.
Right.
And as you said, it doesn't matter how many different ways I arrange the furniture in my living room, we're still going to have that old couch somewhere, and there's never going to be a new couch. And that's exactly what you're saying about the DNA. There are ways that God has made for there to be variety within that species or that kind, but there's no way to have a new kind in there, is there?
Exactly, and that's
the increase in information. Gene duplication is any duplication of a region of DNA that contains a gene. It may occur as an error in homologous recombination, a retrotransposition event, or duplication of an entire chromosome. The second copy of the gene is often free from selective pressure, that is, mutations of it have no deleterious effects to its host organism. Thus it mutates faster than a functional single-copy gene over generations of organisms. Recombination. Recombination is a much more powerful way for DNA to change. If an organism's genome were written out as text, mutations would be single-letter mistakes, whereas recombination takes whole words, sentences, paragraphs, pages or groups of pages and moves them to different locations. These new locations could be elsewhere in the same paragraph, page, bookshelf, or library. Obviously, a powerful mechanism like recombination should be incorporated into anyone's understanding of the theory of evolution. There are three kinds of recombination. The transfer of DNA across species boundaries. While recombination moves whole blocks of genetic instructions within a cell, other processes move whole blocks of genetic information from one bacterium to another bacterium of a different kind. In the analogy between genes and written text, this move is a transfer of paragraphs or pages from one library to another. One such process is transformation. Here pieces of genetic instructions are released by a bacterium into its environment. Another bacterium, not necessarily of the same strain, picks up the DNA and incorporates it into its own genome. Conjugation is the bacterial version of sex. In conjugation, bacterial cells actually connect, and the male donates a piece of DNA to the female. Transduction is yet another way for bacteria to exchange genetic material. In transduction, a virus takes up a piece of DNA from its bacterial host and incorporates it into its own viral genome. After the virus has multiplied, many copies of the virus erupt from the infected cell. Depending on the kind of transduction, some or all of the daughter viruses take copies of parts of the bacterial DNA with them. When one of them infects a new cell, it inserts the stolen DNA into the new cell, where the stolen piece becomes integrated into the new cell's DNA. The stolen piece may be a whole gene with which the cell acquires a new function, as was reported in June 1996 by two scientists at Harvard Medical School. Transduction by viruses works in eukaryotic organisms as well. The discovery that large blocks of genetic instructions can be swapped and transferred among creatures is a clue that the insertion of new genes could be the mechanism behind evolution. If viruses can transfer eukaryotic genes across species boundaries, and can install their own genes into their hosts, the case for the new mechanism is even stronger. As we will see, viruses do just that. This method of acquiring genes is not in doubt. Among bacteria, for example, there are some well-documented cases of homologies between viral genes and their host counterparts. Some past exchanges have occurred between distantly related phages and between phage and host. Eukaryotes are also known to acquire viral genes, and the phenomenon is not rare. Endogenous retroviruses and retroviral elements have been found in all vertebrates. In conclusion, viruses could easily provide a way for new genes never before encountered by a species to become part of its genome. That viruses install new genes into their hosts is not speculative, it is a well-known fact. That transferred genes are important in evolution is becoming well established. According to cosmic ancestry, the horizontal transfer of genes by viruses and other means is essential for evolutionary progress. It is almost certainly the case that some modern-day retrotransposons are derived from retroviruses that lost their infectivity and are more properly considered as ancient endogenous retroviruses. J.D. Boak and J.P. Stoy, 1997.
Unlike you and I, or any other creature you can see here at the fair, microbes can pass their genes not only to their offspring, but also laterally to other microbes of the same generation. Phage conversion can result from transduction. Genetic material passes from one bacterium to another via a bacterial virus called a bacteriophage. The virus injects its DNA into the bacterium. The bacterium then becomes a virus factory. replicating the viral genome, making viral coat proteins, and eventually releasing hundreds of new bacteriophage.
Every once in a while, during replication of the viral genome, a small piece of bacterial DNA gets inserted into the coat protein as well. And the live virus now transfers this genetic information from the recipient bacteria to a new bacterium.
In conjugation, another form of genetic transfer, a donor cell comes in contact with a recipient cell by way of a conjugation tube known as the sex pilus. When contact is made, a single strand of DNA containing the genetic information goes through the pilus from the donor cell to the recipient cell. A second strand of the genetic information quickly gets synthesized in both the donor and the recipient cell.
So ultimately, both cells now have complete copies of the genetic information that was transferred.
So what's the significance of that from the perspective of someone studying disease, such as yourself?
Well, we now know that a lot of genes coding for drug resistance, antibiotic resistance, reside on plasmids. So a single bacterium in a population harboring a conjugated plasmid containing an antibiotic resistance gene can ultimately spread this genetic trait through the entire population of bacteria. And this could happen quite quickly in the lab.
In transformation, a wide variety of bacteria have the ability to bind free-floating DNA from their immediate surroundings. This DNA may then be inserted into the chromosome and become a piece of permanent genetic information. Bacteria that acquire DNA in this manner inherit the genetic traits that are encoded in the incoming DNA.